Hello, everyone, and a very warm welcome to you all, and thank you for joining us here today. My name is John Norman. I'm Head of Marketing at Optibrium and moderator for this webinar, Predicting Pharmacokinetics Parameters and Curves. Okay, so now it's time to hand over to our presenters, who today are Matthew Seagull, CEO at Optibrium, who will be our host for today's webinar, and our guests, Tom Whitehead from Intellegens and Nigel Greene from AstraZeneca. Matt, I will now hand things over to you.
Thank you very much, John, for introducing the webinar, and thanks to everyone for joining us today. It's so good to see so many of you in the audience and lots of familiar as well as new faces. I'm delighted to be joined today by Nigel and Tom. First, let me introduce you to Nigel Greene. Nigel heads the Data Science and Artificial Intelligence Department in Drug Safety and Metabolism at AstraZeneca, and is interested in the application of artificial intelligence methods to understand mechanisms of drug-induced toxicity and their translation to a clinical patient population. Previously, he was head of the Predicted Compound ADME and Safety Group at AstraZeneca, and Nigel also spent 14 years at Pfizer, where he started in drug safety R&D and later transitioned to the Compound Safety Prediction Group in medicinal chemistry. In his early career, he worked for LASA, where he pioneered computational toxicology. It's been a great pleasure for us to work with Nigel and all of his team at AstraZeneca. And Nigel, before we get into the main presentation, I know that AstraZeneca is focusing on AI and data science in quite a big way, so maybe you could just give us some background on your overall strategy in the area.
Sure, thank you, Matt. Hopefully you can hear me okay.
Loud and clear, thanks.
Excellent. Just checking I was off mute. Yeah, so as people may be aware, AstraZeneca has really launched into a key strategic objective of bringing data science and artificial intelligence to bear on the whole of drug discovery approaches. And so that's all the way from identification of new targets, all the way through to the design of clinical trials and analysis of clinical trial data. So we're putting a lot of effort into looking at things like machine learning, new techniques for artificial intelligence, and obviously trying to apply that to every single stage across the process.
Fantastic. Thanks very much, Nigel. It's great background, and having set the scene, I'm going to hand over to you now, and be great if you could introduce the project and this specific area of prediction of PK parameters and curves.
Sure. First of all, I'm going to sort of just give a very short overview, and I'm going to actually let Tom talk about a lot of the modeling work that was done. But what I'd like to do is talk about why artificial intelligence and the prediction of pharmacokinetics of drug molecules is so important for us here at AstraZeneca. So what I'm going to do is just quickly introduce the problem, talk a little bit about dose and why it's important in medicinal chemistry, and then obviously talk about how we have traditionally approached prediction of ADME properties for probably the last 20 years, but now, and then move on to talking about how we're now thinking about using artificial intelligence in the prediction of preclinical and human PK. So this slide probably isn't new to a lot of people. There's a lot of challenges in drug discovery, and obviously most of the molecules that we make don't make it as medicines, and there's a number of reasons why that is, but obviously pharmacokinetics is just one of those. So you can either have poor absorption, you don't achieve enough levels in the body to achieve the efficacy that you want. It could be that you, in order to get the drug into the body in sufficient quantities, that you end up getting side effects through off-target pharmacology or other lack of selectivity. And so the pharmacokinetics of a molecule really do play an important role in understanding why a molecule is likely to be successful as you move into the clinic. Whoops. Oh, come on, it's going the wrong way. So one of the things that we obviously look for, and apologies if this is something that is obvious to most of the audience, but having not known who's attending, I figured I'd start at the bottom and hopefully explain drug discovery. But therapeutic index is one of the key drivers that we look for when selecting a molecule. And that therapeutic index really is the multiple of your efficacy concentration, or the plasma levels that you need to achieve to see the beneficial effects of the drug, compared to the plasma levels where you start to see adverse events occurring either in preclinical species or through the study of in vitro assays. So they could be off-target potencies at certain receptors. You could look at things like cytotoxicity or effects in preclinical species. And obviously, as you move into clinical trials, it then becomes looking at where's the maximum tolerated dose. And we all know that really most things in life are toxic at certain doses. That's true of everything. And so really it's about understanding where are you in terms of the toxic concentration, and what concentration do you need to achieve to get the efficacy. And what you want to do is really maximize that differential so that you get the biggest chance of succeeding. And obviously that concentration is often determined by the dose, or is determined by the dose, and it's about achieving a dosing regimen that allows you to go into the clinic with the highest level of success. And obviously dose does make the poison, and there is a statistical relationship between the observed daily dose, or the daily dose of oral medicines, and things like liver failure, for example, and liver transplantation. There's also a link to death caused by liver injury. So, the dose does make a difference when you're going in there. Interesting fact, though, that things like liver enzyme functions are not necessarily statistically significant with dose. There's probably good reasons for that you can see liver enzyme tests going up, but not necessarily inducing injury at certain points. So when you're thinking about the dose of a compound, the dose equation is there at the top of the screen. It's a combination of the average concentration you need to achieve, multiplied by the clearance, multiplied by the dosing regimen, divided by the bioavailability of the drug. And often efficacy is driven at what we call the C trough, or the concentration, the lowest concentration over the dosing period. So that tends to be what you're looking for to have efficacy. However, in order to achieve that C trough, or the lowest concentration you need to do, quite often you have to push the dose to a point where you get a peak in Cmax. And that peak in Cmax is obviously something that drives the off-target pharmacology and can then lead to the adverse effects that you see in the clinic or in the preclinical species. And so lowering clearance helps you to minimize that difference between C trough and Cmax, and therefore you're enabling yourself to be able to go higher in doses in the clinic without necessarily hitting that limit of safety. And obviously this is all defined by the structure of the compound and the properties of the compound, and so inevitably it becomes an important design criteria in medicinal chemistry. So the importance of dose not only determines things like safety, but it also can lead to a lack of success in the clinic. And the dosing regimen becomes important when you're thinking about compliance with medications. And by compliance, I mean that the patient actually remembers to take the drug at the right time, on the right day, during the course of the treatment. And obviously, in certain patient populations, such as the elderly or Alzheimer's patients, memory is particularly important, and keeping a dosing regimen becomes highly critical. And so in a lot of studies, they've seen that actually compliance in clinical trials is with a once-a-day medicine is actually at about 80% of patients. So 80% of the patients are remembering to take the drug at the right time once a day. However, if you go up to twice a day, that drops to about 70%. And then obviously as you go up in more frequent intervals, the more likely it is that patients are going to forget to take one of the tablets at the right time, and therefore they're not going to achieve or have the right concentration of the drug in their bloodstream, and therefore you risk losing efficacy of the drug in the clinical trial. And obviously if you fail to show efficacy in the trial, then your drug is less likely to be successful. And so the dosing regimen is actually very important. And most oral small molecule programs are aiming for once a day, because that seems to have the highest level of compliance in patients. It also minimizes the number of tablets that people have to take. And there is a sad statistic that I think in the US that most people, by the time that they are age 60 or higher, are taking 5 or 6 medications a day. And so having them remember to take 3 or 4 tablets per day instead of 1 tablet per day is actually is going to be even more challenging. And so in essence, the dosing regimen really does dictate whether you're going to be successful. And it also can lead to an increase in cost in medical costs. So having said that, the traditional approaches to ADME that we've probably done in the last sort of 20 years or so is to develop high throughput screening assays that look at things like lipophilicity, permeability, solubility, and clearance. And we use those to select compounds that we believe have the right properties. And obviously with these high throughput assays, we can develop computational models for those, standard QSAR type models, and on these assays, and which can be used to help design novel molecules. We have a lot of data points, typically more than 10,000 data points for each of these assays. And of course, it's very quick and easy to test the model performance by taking the compounds you synthesized and throwing them into the assay and seeing how well you did. However, the challenge here, of course, is that you're actually predicting a model of the ADME endpoint that you're really interested in is what happens when you give a human being the medication. You're not actually predicting the endpoint, you're predicting something that is a surrogate for that. And so that creates some degree of uncertainty when you extrapolate from an in vitro assay to in vivo to a preclinical species, and of course from preclinical to human is a bigger extrapolation again. One of the things that we can do, of course, is develop models for these simple assays. And as you can see here, these 2 models are performing very well. We get pretty good R-squareds for things like log-D and human plasma protein binding. But when we go on to look at things like human liver microsome clearance, for example, the models don't do quite so well. And there are reasons for that. One is that the assay itself has a high degree of experimental error. We also only do a single N of 1 typically when we run these things in high throughput mode. And also the compounds are I guess, a little bit dirty when they go through, because it's run automatically as soon as the compound is synthesized. It's recrystallized or purified, but essentially it's perhaps not as clean as you would want it to be. And obviously that introduces a further error in some of the results that you see. And so typically the error on these assays can be as much as 2 or 3-fold. And so on a 2 log unit scale or 2.5 log unit scale, this can actually introduce quite a lot of noise from a modeling perspective. It makes it quite challenging to get reasonable models from this. So what we wanted to do was to think about how could we use AI in the prediction of human PK? Because obviously being able to predict this at the point of design gives us a great deal of confidence to move forward with a candidate that has all of the right properties that we wanted to have, and that we would be able to achieve efficacy in the clinic at a dose that is both safe and efficacious. And obviously this helps reduce resources in terms of in vivo testing and also time in synthesis if we can actually design the compounds up front. However, predicting human PK is quite a big challenge. Getting hold of enough data for producing reasonable models is challenging at best, but we wanted to try and push forward, and so we focused on, could we predict the in vivo preclinical data? So could we predict the I guess low dose rodent or rat, in this case PK experiments, where you give 1 to 2 milligrams of drug and you look at I guess the concentration over time, and then you can calculate a number of parameters from it. And then obviously deciding whether that produced better models than going after the in vitro assays will help guide us in how could we then extend our learning into human PK predictions, and perhaps even do some transfer learning between the rodent models and the human models that we wanted to do. So as part of this project, we actually selected about 3,000 compounds. This wasn't our entire data set for rodent PK studies, but these were things that had been run on a consistent format at a single CRO, so that we knew that the data were minimizing the amount of experimental differences between different sites running the same studies. But we had about 3,000 compounds, typically with 2 replicates for the majority of compounds, and obviously 9 to 11 point dose response curves, or time concentration curves, I should say, for the individual compounds. There were 9 PK parameters being calculated for these compounds, so bioavailability, clearance, Cmax, half-life, volume of distribution at steady state, and AUC. But we also had 9 in vitro ADME properties that we used as part of the modeling process. So things like log-D, permeability in the Keiko-2 system, looking at things like human liver microsome intrinsic clearance, using rat hepatocytes to get the species difference between human and rat, thinking about solubility, and obviously thinking about plasma protein binding as well, because all of these properties become important. And we used a variety of different modeling techniques, and this is sort of a summary slide of where we got to. So probably the table on the right-hand side is probably the easiest to read, where some of the coefficient of determinations we got were pretty good for things like clearance, fraction absorbed, and volume of distribution. R-squareds of 0.5 or higher, which is actually pretty good when you think about that the RMSEs for these models were also in line with the experimental error that we were seeing when you looked at the average difference across replicates. And so for clearance, you can see an RMSE of 0.228 compared to an experimental error of 0.25. I think it's unlikely that we would see a great deal of improvement on that model with additional compounds, just because of the amount of experimental error in the system. Things like half-life and the Cmax through IV were actually proved to be fairly difficult, as you can see from the bar charts on the bottom right. We actually had a very difficult time predicting half-life, and a lot of that is due to the fact that we're looking at terminal half-life, where you're trying to measure a very small concentration as your final time point, and that has a huge error associated with it, and also can lead to curve fitting changes which have quite a significant impact on the half-life value that comes out. And so a lot of it is because of there's quite a bit of experimental error and subjective sort of interpretation of the data when doing the curve fit in calculating half-life. So in summary, what we've tried to do here is thinking about safety and ADME. Sometimes I would actually claim that the ADME properties of a compound are as important, if not more important, than the efficacy or the potency of the compound. With the right PK profile, you can often take forward a compound that doesn't necessarily have to be that potent, so long as your PK profile is good enough. You can achieve the concentration you need for efficacy without necessarily having to achieve sub-nanomolar potencies at your primary pharmacology. And more often than not, what you see is that as you drive potency down, your safety, the concentration at which you see safety effects, just comes down with it. And so you're not actually increasing your therapeutic margin by driving potency down. You're actually just keeping it the same, but causing yourself some headaches with the PK of the compound as well. So better prediction of human PK will enable us to select compounds with confidence that our therapeutic index will be maintained. And so this is the ultimate desire in medicinal chemistry, is that when we select a compound as being good enough, that as we move into the preclinical and clinical experiments, that we're actually able to test the hypothesis. So actually, and by that I mean that when we run a clinical trial, if it fails, it's because our understanding of disease was no good, not because we weren't able to dose high enough to test whether that mechanism was actually relevant in that sense. And obviously, needless to say, drug discovery is a multi-parameter problem. and often requires us to think in multiple dimensions, and those multiple dimensions are often more than the average human being can cope with. And so I think the use of artificial intelligence is going to really be a significant advance on our ability to select compounds moving forward. So with that, I'll just finish by acknowledging that this wasn't just me. There was a whole team of people behind it. I haven't put on the help and support from Matt or from Tom on here. These are just the AstraZeneca colleagues that have helped it, and obviously my boss, Stefan Platz, who has kindly given me the free will and the free rein to start investing in these areas. So with that, I think I will hand it over to Tom.
Wonderful. Thank you very much, Nigel. That's a great introduction to the importance of PK, the objectives of the project, and the data that's available. And as you say, we're now going to hand over to Tom. So first, let me just give a few words of introduction. Tom has a PhD in theoretical physics from the University of Cambridge. He's very interested in developing machine learning approaches to previously intractable problems in a variety of different scientific and engineering fields, as you'll hear. And it's been a pleasure to work with him on this project and many others, as well as his colleagues at Intellegens. So, Tom, just before we get started with the discussion of the excellent results from this project, maybe you could just give us a little background on Intellegens and the work that you're doing there.
Yeah, sure. So thanks for the introduction, Matt. So Intellegens is a company that span out of the University of Cambridge about 3 years ago now. We span out of the physics department, where we'd been developing a machine learning algorithm for use with sparse and noisy experimental data, originally in collaboration with Rolls-Royce, where we were developing a new nickel-based superalloy for use in Rolls-Royce's jet engine turbine blades. And we've since gone on to use the same technology, the same approach to sparse and noisy experimental data in a whole variety of different fields. So yeah, starting in materials design, metals discovery, more recently in things like industrial chemistry, whole variety of different fields, battery development, thermometry, basically anything you can think of where there's experimental data available. And obviously we've put a lot of effort now into our drug discovery campaign, working very closely with you guys at Optibrium. And it's great to talk about one particular example of that today.
Thanks, Tom. So it's now over to you to tell us more about Alchemite and the application to predicting PK parameters and curves. Thanks a lot.
Awesome, let's get going. So yeah, I'm going to talk about a bit more about the project that Nigel mentioned a couple of minutes ago. So let's start off by just recapping the project in particular. Nigel went through this. So around 3,000 compounds, each with at least a couple of replicates. For each of these, we were trying to predict a few different properties. So some of these were pharmacokinetic parameters. So as Nigel described, these are sort of single numbers that capture the components of the pharmacokinetic profile. Separately to those parameters, we were also looking at modeling concentration time curves directly. So instead of these parameters, looking at the time variance of the concentration of the drug in the bloodstream. And as well as trying to model these two outputs, pharmacokinetic parameters or curves, we were provided with as input some in vitro ADME properties that Nigel also described, along with some corresponding in silico models that we'll discuss a little bit more down the line. So the machine learning that we were using for this project is Intellegens' software called Alchemite. So what Alchemite does, and some of you on the call might be familiar with this, is imputation. So imputation is the idea of taking a sparse data set, a data set that's got gaps in, where not all of the experiments have been run yet, and filling them in with computational predictions that accurately represent what would be seen if this experiment was run in real life in the lab. So in this particular project, we can imagine having a table of data on the left of screen here, where each row in this table is one of these compounds, one of these 3,000 times 2 and a bit compounds. For each of those compounds, we can calculate a set of chemical descriptors that describe the structure, the chemical composition of that compound. And those are complete. We always know those. We've got this in vitro data that is sparse. So not every one of these in vitro properties has been measured for every compound. And one of Alchemite's strengths is its ability to handle this sparse data as input, the ability to work with data that may or may not actually be present for any given compound. And then the outputs that we're interested in are these pharmacokinetic parameters or curves. So the normal way we might use Alchemite would be to fill in the gaps in a sparse data set. But for this project, we're actually interested in using Alchemite in a different way, which is to predict these pharmacokinetic parameters for new compounds, for looking forward prospectively, as though these compounds have not yet been experimentally measured. So in that case, we've got these chemical descriptors, because if we know the chemical structure, we can calculate those. We may or may not have some access to some in vitro data that's been measured on them, but we don't yet have any in vivo PK measurements. And so what we're going to try and do is calculate them, starting from the ground up, starting from these descriptors, the in vitro data if it's available, and predict all of the pharmacokinetic parameters. For those of you who are familiar with Alchemite, the model is trained in the usual way. It's trained exactly as an imputation model, except that we tell it not to expect any experimental data, any pharmacokinetics data here at test time when we come to run it. So one of the strengths of Alchemite, as we just described, is the ability to handle sparse data, sparse experimental data. Another strength is the ability to handle noisy data. And as Nigel mentioned, noise in drug discovery data is everywhere. So instead of predicting a single value for each of these pharmacokinetic parameters, instead of saying the half-life is 2 hours, Alchemite predicts a full probability distribution of results for each parameter. So sometimes that probability distribution will look like the one on the left of screen here, where it's a very sharply peaked, a very narrow probability distribution. And Alchemite's saying that it's quite confident in its prediction of the parameter here. So it might be 2 hours plus or minus 10 minutes, pretty close. But on the right-hand side, we might have a broader, flatter probability distribution, where effectively Alchemite doesn't know what's going on. It's making a prediction that on average, the parameter, say, is 2 hours, but that might be 1 hour, that might be 3 hours. And so what we should do is probably not put as much trust in that type of prediction. Instead, focus on the ones like those on the left, where we can be confident in the result that's coming out. We should take decisions based on the confident predictions that are more likely to be accurate, therefore more likely to be useful. So just to summarize again where this project was going, we're trying to make these virtual predictions of pharmacokinetic parameters. We were doing this for the latest compounds in the dataset. So we split off, or Nigel's colleagues at AstraZeneca split off the 300 odd most recent compounds, and we were running a temporally split test. This is trying to be representative of what would happen in a real discovery type project, where obviously the later compounds may explore a different chemical space than the earlier ones. So we're making two different types of predictions for these PK parameters. We made two different types of Alchemite model. The first was using just the chemical descriptors. So this is a fully virtual model. It's perhaps pretending that the model, that the chemical hasn't been synthesized yet, that it's just an idea in a computer or in a scientist's head, and we're going to test what we think its pharmacokinetic properties might be. And then separately to that, we supplement those descriptors with the in vitro data and some in silico AstraZeneca QSAR models. So this is a sort of semi-virtual, half-virtual model where the compound has been synthesized and tested in vitro, but perhaps hasn't yet been run in vivo. And again, if we can make good predictions using either just the descriptors or the descriptors and the cheaper in vitro experiments, we can reduce the number of in vivo experiments that need to be run, obviously saves time, saves money, and would be an all-round a good thing. So in this project, as I say, we were training models using Alchemite. We also compared to a variety of different AstraZeneca in-house methods that Nigel briefly mentioned. And then finally, I'll just finish off by discussing some predictions, direct predictions of pharmacokinetic curves, again, for these latest compounds. So the first thing when we start a new project, a new machine learning project, is to take a look at the data and see if there are any observations that we can draw from it. So here what we did is generated a correlation matrix between the in silico, in vitro, and PK parameter data that we had available in the training set. So in this correlation matrix, strongly correlated variables are colored red, and things that have got no correlation between them are colored blue. So the first thing that we can notice is that on the right-hand side of the screen here, the top right boxes show that there's a weak correlation between the PK parameters and the in silico and in vitro data that's available. This means that a simple linear model is probably not going to do very well here. We're going to need a more powerful machine learning technique, like some of the ones that we'll discuss in a second. So another thing that we can note is that there are some strong correlations in this data set between the in vitro data and the in silico results. We think that this might be a property of this data set due to the in vitro data being in the training set, these compounds being in the training set for the in silico models, which perhaps gives a slightly over-optimistic view of the quality of these models. But this does show that it's possible to make good QSAR models for this in vitro data. So once we've got that data in place, we can go and start making some models. So we made an Alchemite model using just the descriptors. So this is the fully virtual results that I described a second ago. We're taking the chemical structure and attempting to predict the PK parameters directly. Once we've done that, we can measure how well we did using coefficient of determination, which is one of the various R-squareds that get thrown around. For this R-squared, a value of 1 is a perfect model, so we'll never quite get to perfection, but we can aim. A value of 0 is no better than random. And this metric's actually unbound from below, so we can go down to -infinity, which is a really bad model, and we try and avoid those. Luckily, we didn't get too many in this project. So on this metric, we can plot the results here for each of the 9 pharmacokinetic parameters that we were modeling. And we can see that if we look at the average R-squared is 0.25 or 0.3, somewhere around there. It's okay, I guess. It's on the verge of being a usefully predictive model. But we can see there is a bit of variance in that. In particular, the oral half-life, which is the column second in from the left, is quite poorly predicted by these models. And as Nigel mentioned, this is because this is a complicated variable that we're trying to model. And in general, we can see that the oral PK parameters, which are the first 4 on the left of the graph here, are predicted not as well as the intravenous dosing parameters on the right. Again, this is because oral dosing is more complicated and therefore more difficult. So these are the headline figures. We can dive into them in a bit more detail. So if I pick just one of these PK parameters, pretty much at random, the intravenous dosing area under the curve, we can look at how far each of those predictions is from the true value in the test set. And that's measured using, for example, the root mean squared deviation, which is on the vertical axis here. And if we look at the right-hand side of this graph, if we compare our predictions against the true values for all 300 and some compounds that were in the test set, looking at 100% of the data in the test set, the root mean square deviation is about 0.4 in these units. But then as we move to the left on this graph, we can focus on just the most confident predictions. So the ones where Alchemite's error bar is smallest, that probability distribution discussed a few minutes ago is tightest and narrowest. And as we do that, we make fewer predictions. We start discarding some of Alchemite's predictions, but those that we keep get more accurate. And by the time we're predicting just the 10 or 20% most confident compounds, the root mean square deviation has halved, and we're down, getting close to the experimental uncertainty on these values. So this is really good news for the Alchemite method. The most confident predictions are generally the most accurate, and that means we can trust these confidence levels. This also means you can set a threshold at which you're confident taking a value forward. You can say that I'm not interested in a prediction if the uncertainty is bigger than a certain value. So this is one way to increase the accuracy of the predictions, if you like, to focus just on those that we're most confident in and which are most accurate. Obviously, we would like to make more accurate predictions for all of the compounds. And so one way we can do that is by adding more data. So we can supplement just the descriptors with the in silico and in vitro experimental data. And if we do that, the black bars here show that the accuracy jumps right up. Instead of an average R squared of 0.2, 0.3, it's now more like 0.4, poking towards 0.5. And that's really good. That's making it so that these models are usefully predictive. They're able to be taken forwards with confidence to make decisions based on. Again, it's not quite an even split of accuracies. The oral half-life is still not doing so well, but across the board, it's a significant improvement. So those were the Alchemite results. As I mentioned, and as Nigel mentioned earlier, AstraZeneca, in parallel to this, were also generating a set of models on the same data to see how different methods do, how they answer these same questions. So one of the methods that AstraZeneca used is a deep graph convolutional neural network called ChemProp, which came out a year or so ago. This takes a description of a chemical as a graph and extracts information from that that's amenable for use in a neural network. Another approach that was used are things called support vector machines, which are a way of mapping a high-dimensional complicated space to a lower dimensional or simpler to segregate data space. Another approach are gradient boosting trees, gradient boosting regression. This is making effectively a decision tree for your data set, and then looking at where that decision tree is inaccurate and making another decision tree to correct the first one's inaccuracies, then making a third to correct the inaccuracies of the second, etc., until you've got a good predictive model. And then the last method that was used were Gaussian processes. So these are effectively fitting Gaussian models to your data points. These can be very accurate, especially for small data sets, and they can give good uncertainty quantification, although their scaling as you get to larger data sets can leave a little bit to be desired sometimes. And so in this project, we compared AstraZeneca's results using all of these different methods with Alchemite's. And what we saw is that across the board, all of the results, all of the methods are giving fairly similar results. So different methods do better on different endpoints, different pharmacokinetic parameters, but across the board, all of them are doing fairly similarly. This is quite good news for Alchemite. This is using Alchemite in a different way than it's perhaps was originally designed for, making these predictions rather than doing imputations. But it's also a good news that all of these different methods, if you give them the same information, will get to similar results. And they're all extracting as much as they can from the same data. So that's the pharmacokinetic parameters, making predictions for those. As I said at the beginning, we also made direct predictions of pharmacokinetic curves. So there are a couple of things to think about here. So instead of generating predictions for just a set of numbers, we're now trying to predict a whole object, this sort of 1D line through space. And this is more complicated than predicting a 0D number. There are a couple of things we need to take account of. For example, if for different compounds, the concentration was measured at different times, say sometimes it was at 4 hours, sometimes it was at 6 hours, we need to make sure that the algorithm can cope with this. If different compounds had different numbers of measurements taken, we need to make sure that doesn't bias the algorithm. And Alchemite takes care of this all automatically behind the scenes. So what we did for this project was we made a set of predictions for these 300 test set compounds. And then we can plot graphs like the one on the screen now, where the experimental values are shown in blue. And there are 2 lines here because there were 2 replicates for this compound. And we can also show on the same axis the Alchemite predictions in black. We can then compare the two, calculate coefficient of determination, average it across the replicates. And then if we plot out that coefficient of determination for each compound, we get a profile of accuracy. And what we can see here, this is the accuracy for the intravenously dosed results, is that we're actually doing really well. We're finding, I shouldn't be surprised by that, we're finding some really good accuracy predictions for particularly a subset of the compounds. We're getting those R-squared values up near 1, finding really accurate, really precise predictions. We can dive into a couple of examples of this. So if I take an example, let's say from the left-hand side, so a really accurate one, a sort of average, medianly accurate set of predictions in the middle, and then at the end we'll look at one that went less well. So one prediction that went particularly well, this compound here. Here we can see that Alchemite has captured the shape of the curve correctly. It's got the slope, it's got the curvature correct. And in fact, it seems to be sort of jumping around between the replicates almost as we get to the higher times on the right of the graph. So we're very happy with this. Obviously, you'll always statistically get some predictions that work better than others. This is one that went perhaps luckily well, but an average prediction, this is the compound that had the median accuracy, has also gone really well. The experimental results and Alchemite's predictions look really similar, particularly at short times. Again, we're getting a really good fit there from the Alchemite model to the real experimental data. At later times, sort of 2 hours up to 8 hours, there's a bit more deviation between the true values and the predictions. Alchemite is perhaps predicting that the concentration will drop off quicker than it actually did. But what we can note is that the error bars on the Alchemite predictions do cover the experimental data. Alchemite told us that the true result that came out was a possibility, was actually a reasonably probable possibility to come out from the model. And then just for fun, we can also look at one of the compounds that wasn't predicted very well. So on the right-hand side of the screen here, we've got a compound that was predicted with an R-squared accuracy of -16. It's done pretty poorly. It's thought that the drop-off in concentration would be much, much quicker than it actually was. But again, we can note that the error bars here are really quite large, especially towards the high times, the later times. And so perhaps we'd hope that in some triaging, some post-processing of these predictions, that maybe this prediction wouldn't be included. In fact, wouldn't be included, because the error bars are large, the confidence in this prediction is low, and therefore we shouldn't trust it and take it forwards. So these were results on intravenous dosing. We did also carry out the same analysis for oral dosing. As we discussed earlier, this is a bit more complicated. It's a much more difficult problem to address. But again, we do find some compounds like that on the left of screen here, where Alchemite does make good predictions. It's getting the sort of up and over shape of the concentration over time correct for this compound on the left of screen. But the error bars are a little bit wide, showing that Alchemite's still not quite sure it's got it right. And on the right of screen, we can see a compound where the error bars are very large. Alchemite is very sure, or very unsure about what's going on, very sure that it doesn't know what's going to happen. And indeed, it's made not the best predictions. But we can also note that there's quite a large variance in the experimental results as well. The two replicates here give quite different experimental profiles. So just to summarize quickly, Alchemite has been successfully able to combine descriptors, chemical descriptors, in silico and in vitro data, and use those to predict pharmacokinetic parameters and pharmacokinetic curves directly. These results were comparable to the best results we found in the literature, though we didn't discuss that today, and also a variety of in-house AstraZeneca methods. The direct prediction of the in vitro dosing curves, pharmacokinetic curves, was particularly accurate, particularly exciting, and this is something that would be really interesting to look into in more detail. And I think the top-level summary is that in silico data is adding value here. It's enabling us to make confident predictions that we can accurately rely on and take forwards to make decisions based on, which is one of the main aims in our field. So with that, I will pass back to Matt, and thank you for your time.
Thank you very much, Tom. That was a great presentation. As many of you know, we've been working with Intellegens for a couple of years now, and Alchemite is really forming an integral part of our augmented chemistry software and services. As you can, well, you probably can't see, we've got a deluge of questions coming in, which is fantastic. But just to give you a little bit more time to enter your questions, I thought I would just tell you in a couple of slides how you can actually access Alchemite and work with us and use this method on your own projects. So first of all, as Tom mentioned, we have an exclusive partnership with Intellegens. So Optibrium and Intellegens have been working very closely together now for over 2 years to apply Alchemite to a whole range of different challenges for modeling in drug discovery. This has resulted in several papers. A few of them are listed here, but if you go to our website, you'll see many examples of a wide variety of different applications. We offer access to Alchemite, first of all, through collaborations, very similar to the one that you've heard about today with AstraZeneca. We can look at applications including sort of filling in the gaps in your database with confident results and targeting high-quality compounds, identifying the most important data to generate and help to prioritize experimental resources. It can also be used for virtual screening as well, looking at target activities, ADME physicochemical properties, and helping to find high-quality starting points or new optimization strategies. If you're interested in applications like that, we have many different webinars on our website, and as I say, some papers now are really going into a lot of detail. And the way that we work is to discuss very carefully with you the data that you have, the objectives that you want, and then we'll discuss how and if for Alchemite can be applied to address those challenges and provide a sort of a tailored plan and deliverables and so on. So if you're interested in learning more about that, you can contact us at info@optibrium.com. And most recently, in fact, just last week, we announced the launch of our new software platform called Cerella, which enables deployment of Alchemite for active learning in the context of drug discovery. We gave several different examples to show how this can extract more value from your compound data, confidently target high-quality compounds, and accelerate drug discovery cycles. This connects directly with your data sources to extract the latest data with which to build Alchemite models, to impute and fill in the gaps. On the right-hand side here, you can see in the data that has not yet been measured for your compounds, and to make virtual predictions. And the results of those and those models are available through our Stardrop software, but also for any platform that you wish to analyze, visualize, and work with that data via sort of industry standard interfaces. So if you want to know more about that, you can watch our webinar that was just last week. There's a link there, or visit our website, and we've got lots of examples in much more detail about our Cerella software. And so without further ado, as John mentioned at the beginning, you're very welcome to type in any questions that you may have in the Q&A panel in the GoToMeeting controls. And we'll get through as many of those as we can in the remaining 5 or 10 minutes of the webinar. We might go over a little bit. I think there's probably already too many questions to answer all of them in the time. So if we do run out of time, we'll send you an email and follow up afterwards to make sure that you've got the information that you want. So looking through the questions, I can see lots of great questions here. And I think the first one is probably targeted primarily at you, Nigel, actually. And that's talking about some specifics of the data that you have generated and are using as inputs to these models. In fact, there's a couple of questions that really go hand in hand. So I'll read out both of them because I think they sort of blend together. One of them is, what was the consideration behind including human liver microsomal clearance as a descriptor for predicting rat PK parameters? And the second related question was, why use rat hepatocytes and human microsomes to compare species rather than rat and human hepatocytes or rat and human microsomes, if you see what I mean?
Right. So well, first of all, I think part of our high throughput screening platform and the default, what we would call wave 1 assays, includes human liver microsomes and rat hepatocytes. And so those are the assays that we would run routinely on all compounds after synthesis. We do not typically run rat liver microsomes because rat hepatocytes are fairly easy to come by. The reason we don't run human hepatocytes is generally because they're expensive and requires sort of a donor to donate some liver, either, well, sort of getting hold of donor livers is actually quite challenging, and obviously you can get donor to donor variability, and that causes some issues around experimental differences between batches. And so typically we would use human liver microsomes early and perhaps follow up once the compounds look promising with human hepatocytes at that point. However, it's not an automated submission process in that case. So hopefully that's clear. I do take the point about species differences, and I think ultimately we were looking at how we would predict human PK rather than rat. We're not looking to optimize for rat PK, but we felt that human liver microsomes do actually give us some information about how it might behave, generally speaking, in a more CYP enriched environment in that case, so that you get some degree of knowledge around clearance. And there is some correlation, of course, between human and rat.
Thanks, Nigel. That's great. And actually, there's another question here that is probably best directed to you, Nigel. So you reported some results from your modeling specifically for rat clearance, bioavailability, and volume of distribution. And you had a metric for those saying a percentage within 3 log. One of our audience has asked for clarification if you mean percentage of test set compounds within 3-fold of the observed values, or percentage of test set compounds predicted within plus or minus 3 log units of the observed value.
Yes, sorry, that's probably a typo on the slide. So it is actually within 3-fold, not 3 log units.
That's great. Thank you very much. So now another good question here, maybe for you, Tom, which is what kind of chemical or structural descriptors were used for the Alchemite models?
Yep, so that's a great question. So for this particular project, we used Optibrium's star drop descriptors, which I'm sure Matt can comment on in more detail. Alchemite itself is fairly agnostic to the type of descriptors that are used, as long as they're information rich and describe the compound. But yeah, for this project, we used star drop descriptors.
Thanks very much. I can offer a few words of clarification. So these are the descriptors used in our auto modeler module within StarDrop. So they include whole molecule properties like lipophilicity, molecular weight, volume, charge, flexibility, polar surface area, and so on, plus about 300 structural descriptors represented as counts of smarts patterns. So that's just a little bit more detail for the experts in the audience. Another question here, just for clarification, really, which is, what was the proportion of test set compounds compared to the training set for the Alchemite models?
I believe it was 90% train, 10% split. I wouldn't want to swear to that, Nigel, if you remember better.
It's approximately that, yes. So I think we took, it was 3 months worth of compounds, so it was about 369 compounds were left out, I think, if I remember correctly, something like that. So about a 10% split, 10% test set.
Great, thanks, Nigel. Yes, and absolutely, I think it's good to be clear that exactly the same training and test set split was used for the models that Nigel mentioned, built at AstraZeneca, and those that Tom was describing, built with Alchemite as well. So it was a like-for-like comparison, and it was a temporal split. So as Tom mentioned, it was the most recent sort of 300 compounds that were used as the test set. So here's another great question, and something I think we're looking at very carefully, which is, do we predict the PK parameters directly, or do we predict the PK curves and then calculate the PK parameters? So maybe I'll start with you, Tom, and Nigel might like to comment on that as well.
Yeah, I think someone there has been reading our future research plans. So for this project, the results we presented here, for the PK parameters, we predicted the PK parameters directly. We didn't go via the curve. It's definitely something that's really interesting to look at, whether once we've predicted the curve and then extract the parameters from that, whether they're consistent, whether they're the same. And we've done some first steps on that, which maybe Nigel can describe, and definitely interested in doing more. Yeah,
obviously the prediction or comparing predictions of parameters from curves means that you have to use the same curve fitting approaches in both the, I guess, the training set and the, or I should say, in the actual data versus the predicted data. And that presents a few challenges in that more often than not, it's a somewhat subjective curve fit, particularly as I mentioned earlier with the last data point. So there have been some challenges with using the predicted curves to calculate the parameters. We are finding that it's not really providing a great deal of improvement on the predicted performance at this point, but the jury is still out a little bit. We need to do a bit more work on that.
Thank you very much. So another good one here. So was there any exploration of building Alchemite models or other model types just with the in vitro data as descriptors? I think, Tom, you looked at a number of different combinations. I don't know if that was one of them. Maybe you can comment there.
I believe I didn't look at using just the in vitro. I looked at using the descriptors and the in vitro, but not the in silico models, which we didn't discuss too much, but were based on that same in vitro data. I don't know, Nigel, if you've looked at that separately, just using the in vitro as input, but we found that the sort of limited correlations between the in vitro data and the PK parameters meant that it was good to have access to information about the chemical structure as well.
Yeah, so just to expand on that, I don't believe we looked at just using the in vitro data, and there was a good reason for that, in that the intent was to be able to use the model at design rather than at the point of synthesis. So you would want to be able to, or after the compound synthesized, I should say. So we would want to be able to predict from just the chemical structure if possible. And that's why we didn't go down the route of looking at just the in vitro data as descriptors.
Thank you very much. Gosh, I don't think I've ever seen this many questions for a webinar, and I'm aware that we're already well over time. So maybe we'll do just a couple more before we finish, but we will definitely follow up with everybody who's asked a question to provide the information that you want. But here's another great question. I don't think it's one we've looked at, so maybe Nigel, you and your team have. But the question is, it looks like the predictions are better for higher clearance, shorter half-life compounds. Is this the general trend?
I'm not sure I can answer that question. I don't think we've actually looked at it in that way. I mean, the objective was to, in essence, remove or deprioritize compounds that we thought would have higher clearance, or clearance beyond liver blood flow, which is essentially a compound that's not really going to be a very useful compound at all. I can comment that in terms of our utility of the model, the early data suggests that we have actually managed to reduce the numbers of compounds that have high clearance by about 50%. And by high clearance, I mean greater than liver blood flow, which is really what you want. So in using the model proactively to select compounds, at least based on the few months data that we have, we've actually reduced that amount by 56%. So it's actually having a significant impact on compound selection that goes into an in vivo study. But I don't think we've actually looked at model performance in the different ranges of clearance and half-life.
That's a great result, though, and yeah, it'd be really interesting to look at. Like with all the best questions, we'll have to look at that one in the future. And then I think perhaps to sort of wrap up, here's another great question. For the imputation method, is there a lower limit to the sparse data set? I.e., what is the minimum number of useful data points? I guess that one's for you, Tom.
So I guess the theoretical minimum on getting something interesting out is probably 1 or 2 data points per assay. Obviously, we're not going to get something incredibly useful out of that. I think we've found useful predictive models with between 10 and 20 experimental measurements per endpoint. So 10 or 20 compounds that have been measured in a particular assay. And then if there are several assays which perhaps have overlapping, not identical compounds that have been measured in them, we're able to leverage those correlations. So I think that's the sort of ballpark, that's the lower limit for the number of compounds that we need to have experimental data on in order to get something interesting out. It then scales right up. Obviously, if you're very lucky and work for a global pharmaceutical company and have large amounts of data, we've run this also on millions of compounds data sets. So it goes from the little to the large.
That's great. Thank you very much, Tom and Nigel, for answering the questions. We've got probably twice as many we haven't managed to address in the time as that we've already answered, but we will definitely follow up. And that just remains for me to say thank you very much for joining us today. If you do want some more information, please do contact us at info@optibrium.com. So again, thank you all very much, and we look forward to seeing you at a future webinar. Bye-bye.